Good morning, everyone. Thanks so much for coming here today. I had something written out, and then I've already told this story to our speaker that my car unfortunately was broken into last night, and so I had a different jacket on, and my intro was actually in the jacket. Then I switched quickly to be able to bike here. So I'm going to go off memory, and if I get it right, great. And if I get my intro wrong, then it'll just make you seem even better than you already are. So I'm going to introduce Dr. Marie Schmidt as our Locke lecturer today. She did her undergrad in medical school at NYU, where my father was one of her professors, it turns out, and then did training at Bellevue, did hematology and oncology fellowships, ended up going to Columbia, was on faculty at Columbia for many years, rose up to professor, and then moved over to NYU, where she is the Iven Young Professor of Endocrinology and is the director of the Diabetes Research Center. She has won many awards. She's a member of ASCI. She won the AHA Basic Research Prize about 5 years ago. She received a Mentoring in Women Award from the ATVB Council of the AHA also. And I got to know her a little bit because she was friends with David Dechek. He was the senior associate editor at ATVB. She's the editor-in-chief of that. And when David died about a year or so ago, we had reached out and we had corresponded. And she really felt true loss, is what it felt like. She came and did the Coffee and Cardiology podcast that we hosted that discussed or talked about David, and she started an award through the ATVB Journal of the David Dechek Mid-Career Investigator Award, which I think is just, it's fabulous. And the description that she put together for it talked about his sort of striving of excellence. And I really, when I think about David, that's really one of his defining characteristics to me. So sort of my last thing to say is I think talks like this are so important in our current climate of research in this country and to really recognize the value of basic science research. And so I'm really excited to hear Dr. Marie Schmidt talk today as our Locke lecturer.
Thank you, Jordan. Well, thank you so much, Jordan. Thank you, everyone. Thank you for coming, and it's really nice to be here. Yesterday, I was at the Breakfast Club. It was the Dechek lecture, and it was really, I have to tell you, it was so nice to actually meet Helen because, of course, just like with Jordan, so much correspondence, but it was nice to see her face to face. So I'm very glad for that opportunity. So today, this talk is different. This is more, you know, some of the real nuances of the science that we've struggled with over the years to understand the pathway that we work on, and then some new data, which we are very intrigued about and don't really have answers yet, but kind of as Jordan alluded to, you know, that's the path, that's the life, and we all just, you know, keep going. So when I moved to Columbia as a postdoc after my clinical training, the essence of the work that I was doing was to understand how products of high glucose called advanced glycation end products that, contrary to essentially mechanisms of direct effects of high glucose, these AGEs, advanced glycation end products, form from irreversible modifications from glucose onto lysine and sometimes arginine residues. And the hypothesis that our lab was working on was that these AGEs may actually interact with cell surface molecules and in that context be able to contribute not only as biomarkers, but as potential mediators of complications. And so the project that I embarked on was to try to identify on cultured endothelial cells if there could be receptors for these AGEs. And it took a few years, a lot of brute force work in terms of binding gels, blots, etc., to identify what turned out to be a newly described member of the immunoglobulin superfamily that we called RAGE. When RAGE was cloned, we identified that it was very typical Ig superfamily. There was one variable followed by two C-type domains, a single transmembrane spanning domain, and a short, highly charged cytoplasmic domain that over the years was shown really by many labs to be critical for downstream signal transduction. Also over the years, the first few years of the discovery of the receptor, we also looked into whether RAGE had putative natural ligands, and in fact, very transformative, at least in our view, was that even in the absence of diabetes, these two classes of molecules that accumulate in inflammation, HMGB1 and multiple members now with the S100 calgranulins, were also signaling ligands for RAGE. And in fact, over the years, the innate immunity groups described this type of family of ligands as damage-associated molecular patterns. And this really is very apt description because of the fact that in different forms of stress, the ligands can form endogenously, but they also can be released by cells that are dying, necrotic, as a means of basically just being available in the extracellular space, thereby having the opportunity to interact with receptors. It's also important to note that the extracellular domains were one of our first evidences of a potential therapeutic approach, because you can generate the extracellular domains. They're soluble, of course, and in that means, I'll show you some very early data that was an important proof of concept for us. Next big step was generating antibodies to RAGE, and in fact, we found that it was expressed on multiple different types of immune cells, also on vascular and endothelial and smooth muscle cells, and very interestingly, multiple other cell types either involved in complications or, as I'll show you, the path we took to understanding the receptor in obesity. And so this soluble RAGE turned out to be one of the first really important tools. Once we had generated the antibodies, a very critical question was, because obviously we were hypothesizing that this molecule was involved in diabetes, and a researcher in Italy, Cipollone, had reached out to us to ask us for the antibody, and he was able to show in carotid endarterectomy sample. Here you see the staining, but in this paper that he published on human tissue, he also performed Western blots and noted that the RAGE expression was higher in the plaques from the diabetic people versus the non-diabetic controls. And just like the studies in the mouse, he too found that in the diabetic lesion, there was more RAGE in vascular cells and also in immune cells, particularly macrophages. So, you know, once you identify something like this, the real question is, so what? You know, is this just an accompanying expression pattern in diabetes, or is there perhaps some mechanistic inference? And so that soluble RAGE that I told you about, we used it. We actually generated a mouse model. At that time, there were two major forms of mouse models of athero and hypercholesterolemia. We chose to use the ApoE knockout, and basically we made them diabetic with streptozotocin. And streptozotocin essentially works by damaging and destroying the pancreatic beta cells. And in that setting, the animal gradually becomes hyperglycemic. And for a good part of a span for an experiment, the hyperglycemia is sustained. And what we did is we generated diabetic ApoE knockout mice. And then as soon as they developed the diabetes, we started the animals on soluble receptor or a vehicle. And we were able to show over multiple cohorts that the administration of the decoy, the soluble RAGE, was able to block the progression of atherosclerosis in the diabetic mice. And it's really important to note that the soluble RAGE had no effect on the glucose levels, suggesting that the mechanism was through a downstream interruption of critical signaling pathways. Now, it turned out that one of those is undoubtedly activation of NF-kappa B, but newer data with respect to the tail binding molecule suggest that it may be much more cell type specific and also beyond NF-kappa B. So this is a big part of the work of our lab right now, because obviously understanding that cascade also offers new opportunities for therapeutics in people with diabetes. And so one of the major questions then was the way that I described that experiment to you, it was essentially to block progression of accelerated atherosclerosis in the diabetic mice. When I arrived back at NYU, I began a collaboration with Dr. Ed Fisher, and there we looked at the regression of atherosclerosis in the context of the RAGE pathway. By that point, we had already generated the homozygous RAGE-null mice, so we had two very nice tools to be able to start to address really important questions. And the way the mouse model is done, it's basically a transplant model. So the aortas from diabetic atherosclerotic mice are then transplanted into normal wild-type animals. So the idea is that the wild-type animals have a normal lipid level, of course, and therefore you're regressing athero through that transplantation. And in this experiment, so these are the donors. Here we used a different atherosclerotic mouse, but they all had diabetes, so that you had the donor, which was LDL receptor knockout mice with STZ diabetes, and then the two recipients were either wild-type mice that express RAGE or RAGE knockout animals. And what we found was that in the recipient mice, the deletion of the receptor accelerated the atherosclerosis. So this is the lesion area. And what we saw is that compared to the donor, there was really a very significant regression. And one of the critical aspects that what we found is that if we looked at the lesions in both of the recipient groups, there were many less macrophages per lesion area in the knockout recipients. And in order to try to understand the mechanisms, we actually used a mismatch where the donors were of a CD45.2.2 background and the recipients were 45.1. So this way we could track in the macrophages exactly what the source was. And what we discovered very unexpectedly, I have to say, is that one of the key drivers that was reduced in the RAGE deficient macrophages was the interferon response factor 7. So this was the first time actually that the RAGE pathway was directly linked to interferon-dependent signaling. And so we generated macrophages from the two different groups of mice, and we were able to show that when we expose them to these DAMP molecules that are very increased in the knockout serum, the LDL knockout serum, we were able to show that there was, compared to the wild type where IRF7 was greatly upregulated by these factors, this was much reduced in the knockout animals. We then went back to the mice and we looked for plasma concentration of interferon gamma. And in fact, we found that compared to the, so this is the donor, and then compared in the two recipients, the interferon gamma levels were much lower in the knockout. Now, we've just begun really trying to understand the mechanisms, but what's really exciting is that one would think that one of the biggest effects of the IRF7 in these macrophages would be inflammation, but it actually turns out, just based on some very simple bulk RNA sequencing, that IRF7 also has very substantial effects on cholesterol metabolism, such that we saw differences in cholesterol uptake and efflux genes, and also genes involved in lipid synthesis. So how exactly in a diabetic hypercholesterolemic environment, IRF7 can be driving both inflammation and cholesterol metabolism is really a very exciting question. And we are working on that using now animals that we generated that we can conditionally delete IRF7 from, and we're breeding them into animals where we can test the specific role in macrophages. One of the problems with global knockout IRF7 mice is that IRF7 has different functions, good versus bad, in different cell types. So this work, really showed to us the power of the unbiased sequencing, and now we see new links And that's why this work is so intriguing to us of the RAGE pathway to the type 1 interferon response. So this slide just summarizes a lot of the work we have done over the years, largely not even ourselves, but sharing the soluble RAGE, sharing the knockout animals, where we and others showed not only the effects of the receptor in diabetic atherosclerosis, others have looked at heart failure and cardiomyopathy-like models, and as well in stroke, showing that antagonizing the receptor can reduce these complications. In the microvasculature, we and others have shown that the pathway is involved in a diabetic nephropathy in mouse models and as well particularly impaired wound healing. So importantly, again, the knockout and the soluble receptor studies, in no one's hands did they show that directly antagonizing the RAGE could affect the levels of glucose, suggesting that multiple downstream cascades are really involved in the mechanism. I should also say that even though we began with mouse models of type 1 diabetes, we also have looked at type 2 diabetes models and really see similar activities of the RAGE pathway. So in the course of doing the research on complications, it was pointed out to us that, you know, a lot of these inflammatory pathways also may be involved in the pathogenesis of obesity. And a paper was published about 10 years ago now looking at human adipose tissue of people with obesity but without diabetes, and then lean people. And what these researchers showed was that expression of the RAGE and also one of the advanced glycation end product ligands was greatly increased in obesity compared to leanness in humans. And what was very important about that experiment was that not only did they just show the relative change, but they showed that the receptor was expressed not only in the immune cells, but also in vascular cells. Our work and others has also, you know, shown that the adipocytes themselves express the RAGE. And this really was a very intriguing start to new studies in the lab, thinking about, you know, if you're looking at complications, well, the bottom line, do some of the same pathways that contribute to the complications, do they actually get going well before the development of the hyperglycemia? And so we were able to model some of this in animals. The first paper was also published actually about 10 years ago. While those researchers were working on the human tissue, we were into the animals, and we showed that homozygous RAGE null mice fed a 60% high-fat diet were substantially protected from diet-induced obesity. No differences in food intake and no differences in physical activity. But one of the questions was, well, is there something about deleting RAGE that would block the animals from developing obesity? And for that, we turned back to our soluble RAGE experiments, and we initiated soluble RAGE after about 3 weeks on the diet. This is when the animals are beginning to develop the impairment in insulin signaling. And we found that over multiple studies, that the administration of the soluble receptor compared to a vehicle actually prevented the equivalent rise in body mass compared to the control. Again, these animals had no difference in food intake or their physical activity. And what we did is we sent the animals up to a metabolic phenotyping core. And it was very intriguing that what we found was that the mechanism of this protection, and this was done in the knockout, was through an increase in energy expenditure in the animals devoid of RAGE. So the bottom line was that this was identifying a potentially new role for the receptor in energy hoarding that then in deletion was able to be overcome. Now, the problem with that, of course, is that the global knockout does not really explain where that's actually happening. And the answer to that came when we began looking at adipose tissue of the soluble receptor versus the vehicle-treated animals. and we stained the tissue for uncoupling protein 1, UCP1, heavily involved, of course, in thermogenesis, energy expenditure. And we were very surprised to see in the brown adipose tissue striking up regulation of UCP1. So the source, of course, of the UCP1 is the adipocyte. And by that time, we had already generated conditional deletion of RAGE, AGER is the gene name, in adipocytes. And in these animals, we had the animals that were the knockout only in the adipocyte, and then the controls that were expressing the RAGE in all of the tissues with no deletion. We placed those animals on high-fat diet and found that compared with the controls expressing the RAGE in the adipocyte, that the knockouts had a significantly lower increase in the body mass on the high-fat diet. The DEXA scanning and also actual measurements of fat mass showed that although the deletion of RAGE in the adipocyte had no effect on lean mass, the effect was in the adipose fat mass. So you can see here comparing the green versus the red. And here is the indirect calorimetry. And essentially what we were able to show is that the energy expenditure was higher in the adipocyte AGER knockouts. Now, what took us a while to figure out really was how was this happening? Oh, I should also say that the animals were also protected from insulin resistance. It's always a little tricky. We're doing a lot more experiments to try to separate. So if you're less obese, then for all practical purposes, you probably should be more insulin sensitive. So this is like a chicken and the egg story. Yes, they were more insulin sensitive with the adipose knockout, but which came first and the mechanism is really still under investigation. Inflammation has been strongly linked to these particular pathways, but in many ways it just seems too easy and really not very defining of how this could directly affect insulin signaling. So as I had started to note, we did a lot of experiments in cultured adipocytes and ultimately were able to figure out the mechanism. And the mechanism was that these advanced glycation end products in the adipocyte, without changing expression of the beta 1, 2, 3 adrenergic receptors, what happened was that this RAGE activation was able to reduce phosphorylation of multiple substrates of protein kinase A, one of which was hormone-sensitive lipase, which is very important in lipolysis. So in the cells and then moving back to the animals, we were able to show that these like lipolytic pathways were decreased when RAGE was present, and that this then contributed to ineffective use of energy and the lower energy expenditure. We definitely feel that there's a lot more to discover on this, and we're currently really looking into it, doing phosphoproteomic experiments to really understand the full range of phosphorylation of pKa substrates to get a better idea of the comprehensive role of the pathway in regulating energy expenditure. So a major challenge over the years with the RAGE studies was that unlike many similar receptors, the cytoplasmic domain of RAGE does not have endogenous kinase activity. And so it turned out that it was probably binding to mediating molecules in the intracellular space in order to then activate the downstream signaling. So we used a yeast-2 hybrid assay, and essentially the bait in the assay was the tail, the RAGE tail. So we put it into this unbiased assay to see what the tail might actually bind to. And we were very surprised when we discovered a molecule called DIAPH1, and this molecule was not known to really anyone in the laboratory. And it turned out that the molecule is a formin molecule, and it has multiple roles in signaling, particularly the Rho GTPases. It also can regulate gene expression through serum response factors, and very intriguingly, plays roles in actin cytoskeleton rearrangement and cell migration. And RAGE in multiple different cell types, vascular cells is also in immune cells, very importantly can regulate cell migration through the ligands. We were able to obtain mice that were devoid of DIAPH1 because the first very obvious question is, if you have these animals in the diabetic state, what happens to complications? And of course, we haven't looked at the entire gamut, but we do know that deletion of DIAPH1 is very similar to the effects in vivo of deletion of the RAGE. In terms of multiple diabetic complications, they are quite protected from diet-induced obesity and also from ischemia, reperfusion, and hypoxia injury. And it's very interesting that the mechanism of this is at least in part due to the regulation of downstream genes through the serum response factors. So basically it made sense. And from our standpoint, we still are very, always aware that, is this the whole story? Can we really be absolutely sure? And essentially, like everything else in science, just have to keep developing hypotheses to rigorously test those ideas, and then obviously to be able to, be mindful of the results and what they mean. So a very important, really a major step in our project at NYU was the introduction of a collaboration with Dr. Sheckman at Albany, who is a NMR spectroscopist. And Alex was able to show to very precise detail through his NMR spectroscopy work the precise amino acids in the RAGE tail that were responsible for the binding to DIAPH1. And so he identified R5Q6 in the 42 amino acid tail as being responsible for that interaction. And he showed that when these were mutated to alanines, that the interaction was abrogated. And we showed that when the amino acids were mutated to the alanines, that signaling through the ligands did not occur. So these experiments were a very important step, not only in understanding how it works, but really more importantly, understanding hopefully, therapeutic opportunities. Now, some very recent work in the lab that I'm very excited about, because I think it's finally leading us to how this works, emerged when we studied mice devoid of DIAPH1 in atherosclerosis. So here we use that same model that I told you about before, and we bred the LDL receptor knockout mice with mice that were globally devoid of DIAPH1. And in this experiment, we fed the animals for 16 weeks a Western diet, high fat and cholesterol. And we were then at the end, when the mice were sacrificed, we saw that the deletion of the DIAPH1 was quite protective in reducing the atherosclerotic lesion area. So this is the inanimate here shown in this. And here we also did in these animals on phos analysis of the entire aorta. And it was pretty clear to us when we stained them with oil red O that there was a very significant attenuation in the mice that were devoid of DIAPH1. But what surprised us, because we never really saw this, we saw hints of it, but we never could say that we believed that it was a mechanism, was that the levels of cholesterol and triglyceride in the double knockout mice were lower. So this was kind of shocking because it was not something that we ever found using AGER deletion or the soluble RAGE. And in fact, the FPLC separation of the lipid component showed that the greatest reduction in the double knockout was in the pro-atherogenic lipids. And this led us then, because obviously the liver is so important in these pathways, to then do just bulk RNA sequencing of the knockout and the double knockout. And what we found was that multiple pathways related to cholesterol and triglyceride metabolism, as well as biosynthesis, were reduced in the livers of the double knockout versus the single knockout mice. And what was particularly intriguing was that efflux pathways were absolutely not different. So it seemed as though we were looking more at the biosynthesis pathways, and that somehow when DIAPH was deleted, this was being reduced. So we went through really in this publication a very long series of experiments, because frankly, we just couldn't figure it out. And in fact, we then worked in cell models because cell models of hepatocytes could also help us to avoid the complexity of the multiple different cell types in the liver. And eventually what we came to was the understanding that the nuclear translocation of SREBP1, heavily involved in lipid cholesterol metabolism, and the carbohydrate response binding element were looked at. And the only thing that we found in the cells was that when we silenced DIAPH1, that the translocation into the nucleus of SREBP1 was significantly reduced. In our published manuscript, we went through many different hypotheses for what would be the pathways that were causing that, and we couldn't find that any of the classical pathways actually were attenuated. So we went back to the concept about actin cytoskeleton, and very remarkably, about 20 years ago, a paper was published in Endothelial Cells, basically showing that SREBP1 could be reduced through actin cytoskeleton engagement. And lo and behold, not unexpectedly in our cellular model, we were able to show that F-actin, which is the polymerizing form, was actually significantly reduced in these cultured cells upon the silencing of DIAPH1. So then, to close the loop on the hypothesis of whether or not the actin polymerization was important for the nuclear translocation of SREBP1, we treated the cells with latrunculin B, and this agent blocks F-actin polymerization. And in fact, we found that in the two cells with vehicle treatment or with the inhibitor of actin polymerization, that the nuclear translocation of SREBP1 was significantly reduced. So we're very excited about linking back DIAPH through its fundamental role in actin polymerization. And obviously what's clear is that SREBP1 may not be the only target of this. So we're working on designing experiments really to test that idea. What was very exciting, though, when I told you that we had looked at multiple different pathways to try to understand SREBP1 location, we were very surprised to find that a molecule called cofulin-1, the serine phosphorylation of cofulin-1, was dramatically altered in the absence of DIAPH1. We saw this not only in the aorta and the liver itself, but we also could recapitulate this finding in the cells. And essentially, when you have more phosphorylation of cofulin-1, and the function of cofulin-1 is involved in actin polymerization and tethering, what we found was that the phosphorylation of this molecule was much higher in the absence of DIAPH1. And this is a case where the serine 3 phosphorylation actually inactivates the molecule. So the bottom line is that going into the literature, it turns out that this cofulin-1 is incredibly interesting molecule. It actually can migrate into the mitochondria, where it can be involved in generation of oxidative stress and in mitochondrial dysfunction. So work that I have been doing with Dr. Sheckman that has now really been a long-standing collaboration was to show, he has shown that DIAPH1 can bind to cofulin-1 and be involved in part of its activities. So there's a tremendous amount of work to do to understand it, but the bottom line is that we are uncovering that these RAGE ligands bind to different cells. DIAPH1 is an intracellular effector, and beyond just activating signaling, DIAPH1 can actually bind to molecules like cofulin-1 in order to really change dynamics and oxidative stress. And last fall, we published a paper in our lab with Sheckman again, showing that DIAPH1 can actually bind to MFN2. And MFN2 is mitofusin-2, a very important molecule involved in mito functions and also the mito-ER contacts. And so it's established that the distance, the mito-ER contact, is really very important in regulating so many facets of mitochondrial function. And so in studies that we did in human iPSC-derived cardiomyocytes, we were able to show that the mito-ER distance, if you just induce a ischemia in the wild-type DIAPH expressing cells, here we use hypoxia reoxygenation, that this mito-ER contact is actually shortened, that they're closer together, and that when you delete the DIAPH1, you see an increase in the mito-ER distance, suggesting that there is some protection of the DIAPH deletion from increasing that contact. And the reason why that's important is because we were able to show in the published work multiple different downstream consequences of this in terms of injury, mitophagy changes, and also necrosis. And we used a linker. So in this linker, we were able in the DIAPH deleted cells to block that rise in the mito-ER distance. And when we brought them together, lo and behold, what we found is here when we looked at a marker of injury, LDH release in hypoxia reoxygenation, that the linker was able to block the benefit of the DIAPH1 deletion by preventing the rise in the mito-ER contact. And so if you look at many different reviews, and even in our own paper, we looked at a lot of different endpoints, what this mito-ER distance can regulate. And here we're showing that DIAPH-MFN2 can be part of that regulatory network, is not only the contact itself, but its downstream effects on metabolism, oxidative stress, mitophagy and turnover, and also lipid metabolism. So what's so exciting to us is we may have finally figured out how this effect of DIAPH on lipid metabolism may be actually occurring. And so finally, a portion of our lab for many years now has really been looking at the possibility of targeting RAGE in the humans. Now, if you remember the little cartoon, the initial therapeutic approach was targeting the extracellular V domain. And the extracellular V domain was really where most of the ligands of the receptor bind, but the truth is some of the ligands actually bind at those C domains. And a therapeutic approach that was directed against the V domain actually failed to meet endpoints in a phase 3 clinical trial in Alzheimer. Why Alzheimer? Because of the DAMPs. And so the bottom line was we went back and really tried to think, well, gee, how did that happen, right? Because you would hope that the experiments in the animals would have been somewhat informative, for the human. And in fact, what we realized was that even though these ligands are so promiscuous and they can bind not only at V domain, but also at the constant domains, Alex and I decided maybe we should look more closely at the opportunity to block the interaction of the cytoplasmic tail of RAGE with DIAPH1. And so in 2010, we were funded by JDRF, now Breakthrough Type 1D, to develop a screening assay of looking at potential interrupters of RAGE tail DIAPH interaction. And in fact, together our labs collaborated to screen a 59,000 compound library. We came down to 13 hits that really had multiple characteristics that were important to us in terms of binding affinity, and that the molecules needed to bind to RAGE tail, but not to DIAPH1. And so NYU brought a medicinal chemist to our project, and through extensive structure-activity relationship modifications and many in vitro and then ultimately in vivo assays, we identified a chemical probe, RAGE 229, with a nanomolar affinity for the interaction of RAGE to DIAPH1. In our published paper from a few years back, we tested this in mouse models of diabetes. This is in the kidney, where we were able to show in type 1 and type 2 models that the small molecule probe could reduce the damage in the kidney. And importantly, we also looked at wound healing because of the fact that certainly vascular and immune responses are so critical in wound healing And we were able to show that topical administration in male and female mice was able to improve the wound healing in the diabetic animals. And so a big challenge that this project is going to face is the identification of target engagement biomarkers that can be done in cells or in people plasma. And one of the very natural aspects of RAGE is quite fascinating, is that there are natural forms of soluble RAGEs. They're very low levels, but they're largely generated through matrix metalloproteases cleaving the RAGE from the cell surface. And there are hundreds of papers where individual researchers have looked at soluble RAGEs. R&D, we have nothing to do with that, made an ELISA for the measurement of soluble RAGE. And these researchers could show that soluble RAGEs seem to correlate with human disease. The problem was that the soluble RAGEs, sometimes high versus low, and the opposite, was associated with more or less propensity to diseases in which the RAGE ligands accumulated. And so Angela Raucci, who's worked on RAGE and the soluble RAGEs for years, she basically said, well, you know, it's very possible that the reason for that complication of understanding, you know, the relationship to good versus, you know, maladaptive disease pathway could be because we're not looking at the levels of the ligands. So obviously the hypothesis is that the soluble RAGEs can bind to the ligands, and by binding to the ligands, block them from interacting with the cell surface receptor. And so Angela then developed the assay where she looked at the same time in the humans at the soluble RAGE forms as well as the ligands. And here, because she was interested in diabetes, she looked at the advanced glycation end products. And the bottom line is, through very meticulous statistical analysis, what she was able to show is that the ratio between the ages and the soluble RAGEs was much more predictive of survival in patients with type 2 versus only looking at the ages or only looking at the soluble RAGEs. So for us, this could be very important, because one could imagine ultimately a short trial where before and after S-RAGEs and AGEs were looked at in people treated with the antagonist, and then the bottom line would be, does it change in people that appear to respond versus non-responders? So that problem of the target engagement biomarkers may be solved, but obviously that's going to depend on the studies, you know, ultimately in humans. But, you know, it's, for us, it was an important lesson really in, you know, looking at failure and then trying to leverage the failure to, you know, develop a better hypothesis and, you know, try to be able to test that. So that certainly is the future, you know, for the work that we're doing. And, you know, I think the most important thing is that if we're successful and working with many people, you know, throughout the world, we may finally have a handle on disease-modifying therapies for diabetic complications. And much remains to be done and to be tested and to be shown. But, you know, needless to say, very grateful. This is the current and very most recent group of the scientists in the laboratory, which really is a combination of graduate students, postdocs, and more senior scientists, and of course our wonderful technicians that take such good care of the animals and help us to process the large numbers of samples. And also very grateful for long-term collaborations with Ravi Ramasamy and Alex, and also all of our collaborators, not only at NYU, but outside, and the funders. This is our building where we work. In 2018, we were still at NYU, of course, and were asked to all move. Anyone who didn't do cancer had to leave our current past building and then come into what they call the science building. And so this is the science building of this is the East River, and then this building is just right on it. Our lab is on the 6th floor, and it's very similar design to so many of the labs I visited here yesterday, where it's open lab, right? And the idea of sharing and collaboration. So I want to thank you, Jordan, you know, for inviting me to come, and also to the wonderful hospitality of everyone yesterday and today to, visit UW as part of honoring David, but also sharing, what the work that we're doing and how we're trying to attack diabetic complications. So thank you all.
Thank you so much, Anne-Marie, for coming and talking to us today. Do we have questions? April, looks like.
It was a great talk. Very meticulous work. Are there any human analogies? So have there been any human mutations have been found in any of these pathways that have pathology, that have recapitulated some of these pathologies?
Yeah, that's a really good question, and in general, when large-scale studies have been looked at, there is a SNP in the V domain that was associated looking at large cohorts of people. The first paper that came out on that SNP was loss of endothelial function in the lung in aging. And so it actually made a lot of sense because the mutation enhances the interaction of the ligands to the V domain binding site. But other than that, there have not been any that really were very strictly correlated with Mendelian randomization and this type of thing. So unfortunately, what it looks like is that this type of a receptor can just bind to all these different potentially pathological ligands, and that it may not be the receptor itself, but it's those interactions. I know that is a bit of an issue because there are people who believe that unless your, target can survive Mendelian randomization studies, that it actually may not really truly be related. We have taken the approach that, okay, we accept that and, try to do this in a genetic and pharmacological way to try to show it. I'm not sure how much people really believe that on the Mendelian randomization, but, it is a criticism I do get.
Fareed?
That was fantastic talk. As somebody who attended both talks, I learned so much even today. It was
Thank
excellent.
you. I tried very hard to
Yeah.
make them different.
My question is regarding finding druggable targets. As you may know, the Nobel Prize for Chemistry was won by an investigator here last year for designing novel proteins. Do you think that changes how we screen for things? This library of screening things, can one use AI tools now and come up with things to accelerate things? Have you thought about that? And any comments?
Absolutely, and that's part of the problem, quite honestly, of academics trying to do drug screening, because it just takes so long. And then, of course, what you see is all these other advances. So I think you're absolutely right. And, what you're saying is that we really should go back and try to interrogate them using some of those technologies. It's very funny. I got that same comment from Joe Wu at Stanford. And I was like, he actually invited me to his drug development course. I thought, oh boy, Joe Wu is trying to tell me that this is not the way you do things. But I definitely think we should do that. I have to admit that the medicinal chemist is like, well, why would anyone do that? We already found, but I think you're absolutely right.
Right. Can I just ask one more question regarding your drug targeting strategy? So it's usually appealing to do the receptor on the extracellular because you don't have to worry about getting it into the cell. So did you guys have to do any modifications? Because the approach of looking at those two, that you have to come up with something to make it penetrate.
That's right. And it's very interesting that so of course, you only tell the things that you were successful because that's how you got, to the next step. But that was a big criticism in the grants because they were like, well, these PPIs, this is like a brand new story. And so who was particularly adamant that that is going to be overcome was Dr. DeVita, the medicinal chemist, that these protein-protein interactions, obviously these are largely going to be And he was just absolutely determined that, so one of the factors with this is, balancing solubility versus, the efficacy. And in a manuscript that's just almost ready to be submitted, Alex used very, more complex even than the NMR techniques to actually show how the small molecule could bind to the cytoplasmic domain. So I can't say enough about how critical, his collaboration, because if it was just us doing this, the story would be much softer and potentially not believable. He even developed, FRET assays, and also can do the FRET in vivo, in live cells, and to be able to prove that this is binding to the DIAPH. So, I have to say, as I mentioned yesterday as well, the beauty of collaboration and just, being able to work together, unselfishly for so long is really so important. Without that, structural biology and the biophysics, I don't think we would have as compelling a story. So that's one reason why the medicinal chemist who enjoys this multidisciplinary approach, just scratched his head, well, I don't think we need, but I do think we need that. We have to prove this in more than one way.
Yeah, I was going to ask Jennifer, do you know if there are any questions online that we have? Gotcha.
Oh, okay. That's fine.
All right. Well, I'm going to thank you so much for
Oh,
coming
thank
today,
you.
Dr. Schmidt, and for being with us. I'm going to be meeting with a few people here today.
Yes, thank you.
Thanks for coming from New York.
Thank you.